Good morning, class. This is the second of the set of chapters that I had put across for you to listen online. This is chapter 7. This is following the chapter 6, which was about the cell and its structure and components and everything. Chapter 7 is basically the details on the structure of the membrane of the cell and membrane of just about all the organelles, and basically just membrane and what its functions are, what its composition is, what its functions are, what are the different things that it does, and what is the outcome. This is the synopsis of this particular chapter. Now we move on. As usual, we have the second and the third slide, which just gives you briefly what are the different things that you should know about this chapter. So that is what is listed in this. Okay, so we start with talking about what is selective permeability. The word itself says, I am selective, I choose. Permeable is allowed to pass. So basically, membranes choose what they want to let in and what they don't. Okay, this is one feature which is seen in any living membrane. Now, the time before electron microscope, people had not really seen the finer details. They knew cells existed because of the normal microscope. They knew that the cell is made of different components, but they also knew that there are some membranes enclosing it. That is like sort of logical. But what exactly it is composed of, how it is structured, is something which was like left for people to theorize and scientists to try to find out the different possibilities that could exist. So they sort of knew with the chemical extraction and estimation and testing that membranes are composed of lipids, okay, and proteins, and sometimes some carbohydrates. They just did testing and they would know that these are the components that are present in the membrane. And the lipids that are present are phospholipids. They are amphipathic. What this basically means is, if it has got, like for example, the next page will be able to easily explain to us. Okay, so this is your structure of the phospholipid in short, okay, or in very, very layman terms. So this is the place where it has the phosphate group. So it is hydrophilic. This is the part of the molecule which is hydrophilic, capable of reacting and interacting with the aqueous medium. This is the part which has got the fatty acids, which is hydrophobic. Which is sort of repels water. So any molecule, this is an entire molecule, one part of it is hydrophilic, one part of it is hydrophobic. This molecule then is now called amphipathic. Okay. So then there were some Dutch scientists who put forth a membrane model wherein they said that phospholipids are present in a bilayer. And then there was no way they could explain the presence of proteins that they found out when they did a chemical analysis. So the second suggestion was it probably has a protein coating on top. Okay. After some time, they saw different scientists. Now, I don't even want to take different names and talk about it. Different scientists examined different cell membranes. They noted that different membranes showed different functions. And there were proteins in the cytoplasm that were water soluble, but these membrane proteins were amphipathic, only partly soluble. And if they are layered or coated, their hydrophobic parts could be exposed, and it sort of did not satisfactorily explain their presence and how they could be present in the model. Basically the model that was suggested was sort of not really helpful in completely satisfying all the different observations that were made, and it didn't explain all of that. So the best model which fits the findings is the fluid mosaic model. Okay, the scientists went ahead with a process known as a freeze fracture method and tried to find out what is the components of the cell membrane. They elucidated the following, which we will come across in the next slide. Okay, so this part is understood. So you have a layer, lipid bilayer, that's what it's called, because it's got one set of lipids and another set of lipids. So it's a layer of one set of lipids and another one. So a lipid bilayer is present, and this lipid bilayer is arranged in such a way that the hydrophobic heads were exposed to the water on the outside, and the hydrophobic heads are again exposed to the cytoplasmic water on the inside. Okay, and in between them were the hydrophobic tails, which was sort of making sure that not every cellular content is just escapes through the cell and the cell is sort of lost. Okay, it holds the cell together. Now the fluid mosaic model explained the presence of the proteins in the following manner. It said there are proteins present which can be totally through and through the membrane. So they are sort of called the integral proteins. And there can be just proteins present only on the outside or only on the inside, which means they are called peripheral proteins. They are present in the periphery. Okay. So this technique sort of gave them a very good idea of how it is arranged. So they went through electron microscopic technique, wherein they took out the lipid bilayer in two halves, kind of peeled it off, and they looked at the inside of the extracellular layer, okay, and the inside of the cytoplasmic layer, and they sort of could make out all these granular parts are composed of proteins which are more on the inside, because the cell requires a great lot of them to interact with the outside and interact with the rest of the cell components in its entirety, okay, in its totality. Now this picture can be a little overwhelming, so we move to the next slide where I have sort of simplified this picture. Okay, what it means, the picture before or the slide before means is you have your lipid bilayer. It is composed of phospholipids, and it can have a protein which is through and through present, the integral protein, and it can have proteins which is just on the outside periphery or just on the inside periphery. Okay, and they have certain groups, okay, which can react, bind to the microfilaments on the inside and hold it together and interact or hold the extracellular matrix, the ECM that I was talking to you about, on the outside and hold it together in place. Okay, that's what the slide means, simplified version. Okay, then we have the next part wherein I wanted to talk about what could be the composition of the lipids that is present, or the phospholipids that could be present, and what kind of fatty acids it could have, and does it make any sense for it to have the same kind for animals and plants living all over the world, or does it make sense to have a different composition depending on the kind of environment that they are living in. Okay, take the example of a cold environment. If it is a cold environment, okay, and if the membrane has saturated fatty acids, it will solidify. It will become non-functional completely. So it is better for these animals to have some amount of unsaturated fatty acids, which will retain more fluidity. Yeah. Consequently, the animals living in warmer environment, if the membrane has unsaturated fatty acids, it will be liquid completely. There is no structure, no form, and it will become non-functional. So it is better if it has some amount or a greater percentage of saturated fatty acids. Yeah. Then the different proteins that are present determine the function of that particular membrane. Okay. Now, there are proteins called integral proteins which go through and through. They are also present in the hydrophobic layer part. Then there are peripheral proteins which are not embedded, which is loosely bound. There are 6 functions enlisted for the membrane proteins, and they are as follows. There can be proteins which are responsible for transport. They can be channel proteins or carrier proteins which can change shape once they hold on to a molecule. They can change shape and then sort of move or somehow move that particular molecule via a tunnel, via movement across the membrane itself, and release it on the inside and regain the original shape and come back. Okay, like the diagram in the next page will probably explain us better. Okay, so you can have a transport protein which can function as just a tunnel, and an example of that is an aquaporin. Now, aquaporins are responsible for allowing the entry of water molecules into the cell, into and outside. Depending on the number of aquaporins that are present, it is very clear that they are responsible for entry of up to, if like a normal cell which has got normal aquaporins present in it. So they are responsible for allowing up to 3 billion water molecules per second that can pass in a single file through the central channel. If the aquaporin is not present, only a fraction of these molecules can diffuse at the same time in the same area. So you can see how important it is to allow for quicker movement of material. So it sort of facilitates the process. Then you have a transport protein which can change its shape. This transport protein uses the help of ATP and takes in or takes out the molecule with the ATP molecule sort of phosphorylating it, forcing it to change its shape and releasing it out or in, depending on what the situation is. Then there can be enzymes which are sort of arranged in a fashion in the membrane. An example is during respiration, you would come across, we would be talking about the electron transport chain carriers. Now, all of these are responsible for holding different molecules, arranging them in a sort of sequential manner. So you have certain activities taking place, certain conversions taking place. It can also mean if there are certain molecules that are outside the cell and the cell cannot really take it in because it's really too big, these enzymes can be present and on site break it down into smaller sizes which can be taken in. Okay. Then you can have something known as proteins. I had mentioned this when I was talking about protein functions in chapter 5, that there are certain receptors which can accept a signaling molecule or a hormone and then sort of be the first for a series or a cascade of reactions that has taking place in response to that signaling molecule. Then you can have something known as cell-cell recognition, which takes place especially when you can see in blood groups. Sort of it recognizes like, and that is fine. We will understand the importance of this much later. Basically, just to understand that the cell can recognize another cell because of certain structures which are present and because of certain glycoproteins present on the cell membrane which can recognize the self, okay, the presence of being self. Then under cell, chapter cell, we had talked about intercellular joining, wherein we were talking about tight junctions, wherein they were responsible for intercellular joining. Okay. And lastly, there are proteins which are responsible for attachment of the cytoskeleton and the extracellular matrix. So, on the cytoskeleton can be attached to the proteins, and the protein can also recognize the extracellular matrix which is present and hold it together and hold everything together per se. Now, to understand the next part, permeability of anything or selective permeability, okay, what establishes this direction? What decides you should go in or you should go out? What will it be, the reason, or how anything is decided? You couldn't just say, I have something and it just diffuses. No, there is a purpose to it, and there is a reason and how this thing takes place. For this, we understand the simple concept of diffusion. Okay, diffusion, if I say diffusion, I'm sure everybody in the class understands. Diffusion means just moving away of some particles. But there is a certain direction. What decides from here to here it will move, or not from there to here? I have had in the past, when I've had this class, in class, I have had in the past some students saying air currents, wind, and fan or something else can decide this. But more importantly, if all these external factors are not in consideration, if we have just the material that we are talking about, there is a certain way in which this can take place. Simply put, in the absence of other forces, a substance will diffuse from where it is more concentrated to a region where it is less concentrated. It will diffuse down, okay, from high to low. And this process is spontaneous. It does not require any input of energy. It is independent of the concentration gradient of something else. Now, at this point, I realize that I've been using this word called concentration gradient. And this, to some, can stump some people. So I have another explanation or another way that I could explain this. I would like you to understand shades from lightest to the darkest, if you need to understand. So that means it's the concentration of that color, right? So kind of, if I have shades, okay, now in this particular situation, supposing this is the darkest, so it has 100% black color paint, okay. Supposing here it is 90% of that plus 10% of some diluting material, then 80% plus 20, then 70 plus 30, 60 plus 40, then 50 plus 50. This is 40 plus 60 diluent, okay. Then 30 plus 70, 20 plus 80, 10 plus 90. So from being the lightest to the darkest, so then this can be say 5 plus 95. This can be say 2.5 to 97.5, just for our argument's sake. So it can have very little, so it can have more than that, then it can have a little more, then so you can have then it can have a little more than that. Now this is completely covered. So here, if you notice, you have, supposing this is white and this is black, so you can see black particles are very low in number. So low concentration of black to high concentration, or the highest concentration. Now this is a concentration gradient. Now if I say it goes from high to low, something like, for example, in this I was just putting a range of colors. Supposing, say, I would talk about a range: strongest strength of perfume in this area and least amount of perfume in this area. It could go in a gradation or a concentration gradient of from high to low. Do you get it? Now this is a long concentration gradient. Okay. Another way to explain this is, if I have a lump of sugar, if I have put it in, it slowly diffuses. So it's most concentrated at one region, then there is more spreading, and then it completely spreads. So from just being concentrated at this small region at this corner, it sort of diffuses till it reaches an equilibrium. Here is another example of something which is very light in shade to being the darkest. Kind of like you would have these makeup layer, or the base layer that you would put. You sort of fit it according to your shade. So you have these different shades that is available. So you, depending on what kind of makeup you want to put, you sort of add more and you sort of create, right? So the way it is present in your makeup palette, it would be something like this. I hope after this effort you understand what I'm talking about when I say concentration gradient. Now, if there is diffusion of one solute across a membrane, it will move from the region of high, because this is the region the solute is high in concentration, to a region where it is low. It will keep moving till it has reached an equilibrium, till both sides have equal amount of the dye. Okay. Now, if it has two solutes, the movement of one solute is independent of the movement of the other solute. So each solute movement is independent. Yeah. Okay. Now we move on to another aspect of this chapter, which is talking about effect of osmosis on water balance, wherein you have movement of water. What is free water? Okay. And there are different terms in this that you have to understand. What happens if an animal cell which doesn't have a cell wall is exposed to different kinds of solutions, and what happens to a plant cell which is exposed, which has a cell wall which is exposed to different kinds of solutions. Okay. Now, an example in this that they have taken is this is first to explain just the osmosis part of it. We are not even talking about what happens, the fate of animal cells or plant cells. We are just talking about with respect to osmosis. Now, in this case, if you notice, this tube, this U tube that is there, and it has got a division or a membrane. It has less concentration of sugar in it and high concentration of sugar in this side. Okay, so it has less solute but more free water, whereas it has more solute and less free water in comparison. Okay, so the solute will move from high to low. So the solute or the sugar will move in this direction. Okay, and again, and the water is more here than here, so it will move in this direction, the free water. Okay, they will move in such a way that when it reaches an equilibrium, probably this is how it would look, wherein it needed to move so both of them reach equilibrium, the solute and the amount of free water that is present. So this movement of free water from more free water region to less free water region is osmosis. Please note, I keep saying movement of free water, water which is free to move, water which is not bound to a solute. Okay. Now, to understand tonicity and its relationship to osmosis, I have set up a picture to understand this. Okay. Consider 3 beakers. 3 beakers have different kinds of solution. In this it has 1% of a solute, here it has 3% of a solute, here it is just water. Okay, this bag that I have, it's like a dialysis bag, and this bag contains 1% of the same solute. In this case, both of them are having the same concentration of solutes, in which case they are isotonic, which means whatever amount of water goes in, same comes out. Okay, it is equal. The entry and exit is equal. On the other hand, if it is the same dialysis bag containing 1% of that solute is kept in a 3% solute, here in this case this 3% has high solute but less free water, whereas the bag has low solute, more free water. So what happens is the solutes will go in. Solutes will go in from high to low, but the water will come out. So this bag is kept in a hypertonic solution, meaning it is kept in a higher hyper concentration of solute. So it will lose water, shrink, shrivel. On the other hand, if I have this 1% solution and I keep it in just water, water has low of solute, no solute, but high free water. Okay, and this bag again has higher solute, lesser free water. Now people will wonder, the same thing but I am calling it different. This is in comparison to the solution in which it is kept. And that is the important part that you have to remember. Okay, so this solution that this bag is kept in is hypotonic solution, kind of saying, oh, so low concentration of solute. Yeah, everybody? Okay, so this one will gain water and swell. Okay, now we have to talk about the fate of an animal cell when exposed to the three different kinds of solution, and a plant cell when exposed to the same. Okay, I would like to go stepwise in this and talk about first the animal cell, the RBC. They have taken the red blood cell, which you will be doing in the lab, hopefully this time, in the lab experiment. So if the RBC is kept in a hypotonic solution, what happens is water keeps going in, because outside the solute concentration is less but free water is more. So greater concentration of free water, the water keeps going in, so it causes it to swell up like a balloon till it can no longer swell and it bursts. Okay, so the water goes in this situation. When the animal cell is kept in an isotonic solution, you know how a red blood cell looks like. It is sort of looks like a donut, and the same amount of, as much water gets in, the same amount gets out. So the solute concentration and the free water concentration is the same in inside and outside. Okay, so it looks normal, no change at all. On the other hand, if it is kept in a hypertonic solution, wherein the solute concentrations are high but water is low, so the water goes out to make up for that, or try and make up for that, so it shrivels or shrinks. This is what happens to your animal cell. But a lot of different things happen to the plant cell. We'll talk about it shortly. One second. Okay, now in your plant cell, on the other hand, when it is kept in a hypotonic solution, it is able to take up as much, takes in, water goes in, okay, water goes in, but it likes it takes in as much as possible. The reason for that I would like to explain is plants don't have like a solid bone structure. Okay, they sort of depend on the pressure exerted by the water against the cell wall or against the sides of the cell in order for them to stay rigid. Okay, now that is called turgor pressure. Now this pressure that is exerted on it sort of makes the cell swell up, but it is very normal for that cell, and any plant cell exists in this condition under normal conditions. Okay, so remember, the same things happen to it when kept in these different solutions. In hypotonic solution, water will go in. In isotonic solution, water going in and out is equal. Hypertonic solution, water will come out, but how they react to that, what results from that, is varied in a plant cell and an animal cell, and it makes more sense for you to understand and remember that very, very well. Now, when you keep the plant in a isotonic solution, it is sort of flaccid or limp. It will even wilt. Okay, because there is no turgor pressure being exerted against wall of plant cell. Okay. On the other hand, if you take the plant and expose it to a hypertonic solution, kind of like a 5% NaCl solution, say salt solution, it will immediately lose all the water that it has and plasmolyze. This plasmolysis that's taking place still maintains the shape of the cell, whereas here it sort of shrunk and shriveled. The reason why it maintains is because of the cell wall that is present, which is like a cage. So though the cell membrane sort of has shriveled up, okay, the cell wall is still present and it maintains some amount of rigidity, and that is what is plasmolysis. I've sort of put in the same thing in a different set of words for people to understand how it works. Okay. Now this I had mentioned when I was talking about vacuoles in this chapter 7. This is about the contractile vacuole present in paramecium. What it does is, if it's staying in an hypotonic medium, what happens is lots of water gets in, okay, and this water sort of can change the concentration or the tonicity of the cell per se. So the contractile vacuole sort of takes in that water and throws it out, kind of acts like a kidney for the cell. Okay. Now this particular picture will mention or talk about diffusion, but diffusion with help. So far that we just talked about as is osmosis, just movement of free water, or diffusion of solutes in and out. This is diffusion with help or facilitated diffusion. It can be of two types. You can have a channel protein which is going to help in the movement, or you can have a carrier protein which carries that specific molecule to the inside or the outside. Remember, specific molecules have specific carriers. Okay. Now, to understand something which is totally different from this particular concept, I wanted to take the help of the hill diagram that I was mentioning throughout the time when I was doing chapter 8 metabolism with you. Consider a hill that is present here, okay, and water is up the hill. Now this water which is present up the hill can flow down, flows down without help, and this is understandable. You don't question it. That's right. So this process occurs spontaneously without input of energy along the concentration gradient. It is classified as passive transport. Now the two different processes, simple diffusion and facilitated diffusion with transport proteins, take place passively. And it can use a channel protein or carrier protein. That doesn't mean anything. It just means they are helping in the process, but they wouldn't have helped unless the concentration gradient already moved it in that direction. On the other hand, if you have water at the bottom of the hill and if you have to take it up the hill, what happens is this process cannot go up. It can only go through with whatever help that it can get. The help that it gets is, or it needs, is in the form of energy, in the form of ATP. Okay. Now what is ATP? We'll talk, or I think I have already mentioned in chapter 7, so then we'll hold on that. This goes against the concentration gradient. sort of fighting against the current kind of situation. This process is called active transport. It uses the help of transport proteins similar to the facilitated diffusion which uses transport proteins. But these transport proteins require energy in the form of ATP. These guys will just do it anyways without that energy requirement. That's the difference between the facilitated diffusion taking place here and the facilitated help that is happening here, the transport proteins that are used in both cases. Okay, so this picture sort of shows you how diffusion and facilitated diffusion can take place and how they move. The direction in which they move is along the concentration gradient from high to low, right? Whereas in this particular case, if you notice, the movement of this material is, though it is low in this case, it is higher in this region, it still moves in this direction. It goes against concentration gradient. And it uses the help of ATP with a specific protein, transport protein for this process. Okay, so we move on to talk to you how or why does the cell use energy to do this process at all? What is the purpose for doing this? Okay, imagine a cell and it has got low concentrations of certain things. Okay, remember the whole process of diffusion. What happens? You will have them all diffuse till it reaches an equilibrium, and that's it. Stop, right? Once it reaches equilibrium, stop. There is no movement. Now, consider a cell on day 1. It has got less concentration of solutes. So all solutes come inside and it reaches an equilibrium to the outside. Now, can anything else come in? How is the cell going to live? How is it going to acquire all the other materials? It cannot. So, for this purpose, it always strives to keep a difference. Kind of like how I was talking to you in the thermodynamics chapter. It strives to keep a difference. For that, I just want to talk a little bit about what a cell normally looks like, or the dynamics of the cell is like. Supposing this is your membrane. Okay? Now this membrane, outside the cell, it is positively charged. And the amino part is what peeps out from the membrane proteins, which is like plus. Okay? The inside of the cell, which is exposed to the cytoplasm, is negatively charged. The carboxyl part, which sort of keeps the negative charges, is what is present in the peeping proteins that are present. So there is always a difference in charges between the outside and the inside. This makes the cations want to come into the cell and the anions want to go out of the cell. This is sort of helpful. So the constant effort the cell puts is to keep this difference. Okay, there are different ways in which the cells can keep this difference. In an animal cell, it uses what is known as a sodium-potassium pump. Okay, normally, okay, inside a cell, okay, or the sodium is high on the outside and the potassium is high on the inside. In which case the sodium should come in and the potassium should go out, right? But the cell forcibly maintains a gradient. To understand what the sodium-potassium pump is, I want you to remember Snooky. Sodium out and potassium in. This is what the pump tries to keep doing. So in order for you to understand what is happening, 3 sodium is thrown out and 2 potassium is taken in at every point. In which case the cell by itself, by this particular process, it uses ATP for this process. By this process, it sort of maintains a lot of pluses on the outside and minuses on the inside. Okay, this continues to keep a membrane potential across the membrane. This acts like a battery and affects the flow of different materials into the cell. So you understand how a sodium-potassium pump works. Okay, so that is what I had written. Now in a plant cell, it uses something known as an ion pump. That I'll explain in the next slide. Okay. So this one just is a diagram which completes what I was talking to you about. 3 sodium is thrown out and 2 potassium is thrown in. And for this process, it uses up ATP. Okay, this is your sodium potassium pump. Okay. Next, I want to talk about the proton pump. Now, we will be talking about proton pump when I am dealing with chloroplast, the photosynthesis, sorry, and you can understand it a little better as to the process, what happens in that. Now, forcibly on the membrane, there is something known as a proton pump which uses ATP's help and purposely throws out protons. If there is purposely protons thrown out, what would happen is the protons would want to come gushing back in. Okay, so when they come back in, because they are in a higher concentration forcibly maintained outside and a lower concentration because of that situation, because of the proton pump, it diffuses back. When it diffuses back, the co-transporter sucrose is also taken in and it's brought back in. So this diffusion back helps in bringing another material inside. Okay. So the combination of proton pump and co-transport of larger molecules can take place, and it is sort of coupled. This is also used in the treatment of severe diarrhea. Okay. Now, this is done. We move on to talking about bulk transport. By bulk transport, so far we've been talking about transport of ions, transport of smaller molecules, transport of different charged material and gases and so on and so forth. Now, bulk transport would mean a huge macromolecule, a huge protein molecule, or a bacteria itself, or some digested material. So you have different ways in which this can take place. One process is, you have to remember, first and foremost, this is possible in animal cells better than in plant cells, because animal cells, you don't have a cell wall, no cell wall. So they are capable of sort of undulating, modulating their membrane, whereas plant cell is pretty rigid, so it cannot do this process. So exocytosis, endocytosis, the different processes are not possible in a plant cell. Okay, so there are different ways in which this can take place. Now, a vesicle which is coming from the inside, the Golgi or whatever, can fuse with the plasma membrane and transport out a material. Okay, that's called exocytosis, means it will transport it out. Endocytosis is similar to your solid eating phagocytosis that I had a drawing in my last lecture, but I can add it on here in the next page and you can have a look at it. Similar to that, you can take large quantities of liquid, which is called pinocytosis. Then there are sometimes some material which are taken in because there are receptors for it which hold it together, and they are taken in through a vesicle process. Okay, the next picture and the picture after that will sort of help clear up what I'm talking about. Now, this process is exocytosis. So this is your endoplasmic reticulum, this is your Golgi. Okay, so whatever material was being made, it is present here, secretory protein, it is made into a vesicle, pinched off, and it fuses with the membrane and releases it out, secretes that material out. This process was exocytosis. Okay, the exact opposite is, this is a cell which can see, say, a bacteria, for example, or it can be a food particle on the other hand. It can take that and gulp it in, engulf it in, which is called endocytosis. Take it in, and it's also called, if a bacteria, whole bacteria is taken in, it's called phagocytosis. Okay, it's sort of engulfed it in and digests it. So that's how it works. Sometimes a liquid can be taken in this process. It's called pinocytosis. So this is your phagocytosis, which is an entire bacteria, okay, or food material, food particle, or pinocytosis, which is a liquid which is taken in, or a receptor which is present in a certain region which can accept a certain particular huge molecule, and they are brought in inside a coated vesicle. Okay, so even if you remember these two and exocytosis and phagocytosis, that's good enough for me. The last 2-3 pages I have just put together all the definitions that I could think of which can confuse students. So the difference between the two of them, or difference between any of them. Diffusion is the tendency of a particle of any kind to spread evenly in available space. It can be any medium. It can be in water, it can be in gel, it can be in air. Okay. Passive transport is diffusion of molecules across a membrane with no energy requirement down the gradient from high to low concentration. Osmosis is diffusion of water, especially free water, across a selectively permeable membrane. Free water only. Water, if it is bound to a solute, is not considered free. Tonicity, ability of a surrounding solution to cause a cell to gain or lose water. This is always relative. So if you have a cell and a surrounding solution, okay, it depends on how much free water this one has and how much solute it has, how much free water this one has, and depending on that, the movement of water is sort of decided in which direction it will go. Okay, so that decides what is the solution, the tonicity of the solution, which will cause the cell to gain or lose water. Osmoregulation is the control of water balance. Okay, it sort of controls it and maintains water balance in any living system. Facilitated diffusion is movement of charged substances across the membrane, which with the help of a specific protein. It may be passive if no energy is required and it is moving high to low. It may be active if energy is required and that particular protein, with the help of a specific transport protein, and it moves from low to high. Okay. Active transport. The cell uses energy to move a solute against its concentration gradient across a membrane. Okay. Now I've also made a note of a specific website which gives a good overview of pictures. If you want any more clearer pictures than what I have drawn. Otherwise, I think we have finished with this chapter. Now, I really want you to listen to this chapter, the chapter 6 and chapter 7, completely. Go back again and again. Keep the textbook open. Keep a notebook next to you and understand and take down your points. How best you understand, how best you're able to answer the study guide questions, and then come back to me with your doubts in class. Because I have put this effort in making sure this PowerPoint is available with the audio, I will not be repeating this lecture in class. Okay? So it is your responsibility to listen to it and come prepared to the next class for working on an assignment. Thank you.